All right, everyone. So we are going to go over what we're going to be doing on Monday in class as I share my screen. So I gave you the lab that on in class on I believe that was Tuesday. So you have a blank sheet of paper to write the notes that we're going to write right now. All right. So in this lab, we're going to be looking at two problems. Okay. The first problem, okay, is do germinated peas or non-germinated peas perform respiration faster? So that's the first question we're going to be looking at. And then the second question that we're going to be looking at is how does temperature affect respiration? And we're going to be doing only two temperatures. We're going to be looking at room temperature water or room temperature and then ice. So room is usually around 21 degrees Celsius. Ice will be somewhere between like 0 and 5 degrees Celsius usually. So that is the plan for that. So what I would like you to do is for each of these problems, so which one does faster, germinated peas or non-germinated peas in my charger felt? And for the other problem, how does temperature affect respiration? Does it increase the rate or decrease the rate? I would like you to right now, and I'm not going to do it, write a hypothesis for each of those questions. And again, so the two questions are, do germinated peas or non-germinated peas perform respiration faster? And how does temperature affect respiration? To measure respiration, we're going to be measuring O2 consumption. So I'm going to explain how we're going to do that in this video. We have two independent variables. I would like you to identify the two independent variables that we're investigating. And the dependent variable is going to be oxygen consumption. That's what we're measuring to see the rate of respiration. Okay, so the question is, how are we going to do this lab? We're going to use a device known as a respirometer. So a respirometer has a vial like this, and then attached to the vial is a pipette, kind of like pipettes that we used to measure for the enzyme lab with the spectrophotometer and it turning orange. The pipettes are the same ones. And in this this vial, we are going to put the peas. We're also going to put some other things. So I'm going to destroy the peas for right now. And we're going to put this in a tank of water. Okay, and we know the formula for respiration. And I'm forgetting my sixes, but that's okay. As the peas do respiration, again, you should also write some of the things I say. As the peas do respiration, O2 is going to be consumed. So there's oxygen in this vial, and it's going to be, as the peas do respiration, the O2 is going to be consumed, and water is going to move into this vial, into this pipette. And we can measure how much water moves in. But there's a problem. As you produce oxygen, as you consume oxygen, okay, the gas CO2 is going to take its place. So we're going to add into this vial, you're going to see some KOH, which is going to turn this into a solid. All right. So you're going to see in the instructions, we're going to have a cotton ball at the bottom. Okay, and then we're going to put some drops of KOH, and then we're going to put a non-absorbent cotton ball in between, because the KOH can kill the peas. So we're going to have a regular cotton ball. We're going to put some KOH, which is going to turn this into a solid. so that water will move in when oxygen is consumed. And again, we're going to have 4 of these baths. There's going to be 4 groups, I think, and 2 people are going to be doing cold, and 2 groups are going to be doing room temp there. Now, there are some important controls, things that we need to keep the same. Okay, I'm sorry, constants, not controls. We'll talk about the control test in a second. So some important constants that we have is we have to have the number of P's be the same. We have to have the volume be the same. We have to have the amount of KOH be the same. You can say where are they getting sugar from? Peas naturally have sugar in them. Let me think if there are any other constants. The cotton ball needs to be the same cotton ball size, which is kind of like volume. We are also going to do a control test, and this is the important part. We are going to do a control test with just glass beads. Okay. And the purpose of this control test is to see if there are any changes that are occurring. So you should be writing this. I'm not going to write it. You're going to see if there's any, and now the announcements are happening. And I can't pause this because it's a Google Meet. So there's going to be 2 videos. Hold on. So I'm actually not going to pause it. I am going to put it on mute, and then you can fast forward to when I start writing again. Please bring $2 donation and wear your best. All right, so we are back. I apologize for that. So we're going to do a control test, which is going to have no P's, just glass beads. And you're going to watch another video of a man doing this lab, so it makes more sense after you watch this one. Now, the purpose of this control test is there should be no changes. Glass beads don't do respiration. So if something changes, that means like there's other factors affecting the experiment. So we're going to use the glass beads, and I spelled beads wrong, to subtract out any changes that occur. And so to understand what changes might occur, you need to know some gas laws. So there's a gas law that says if you increase the temperature of the environment, so if you increase the temperature of the environment, then the volume of a gas will expand. And if that happens in our experiment, if the temperature changes while we're doing the experiment, the oxygen gas here is going to change and kick water out, and you're going to see the numbers are going to start to become negative. The other thing that could affect a gas is pressure. If you increase the pressure on a system, the volume decreases. And that can happen in the system by like banging into the table and putting pressure into this vial. That's going to make the volume of the oxygen that's in this tube decrease, and then water is going to flow in, not due to respiration, but due to the fact that you're putting pressure on the vial itself. So we're going to control for these possible errors of temperature and pressure changes affecting the volume of the gas moving, the consumption of oxygen, which when you consume oxygen, there's going to be more volume in the tube. We're going to control for these possible changes by having a tube with just glass beads in it, or a vial with just glass beads in it. So now, the last part of your homework as this video is over, and I'm sorry for the delay with the announcements, but I didn't want to have 3 videos, is you're going to watch the second video, okay? And while you're watching the second video, and you're going to get a grade for this, okay? I want you to write down 4 important designs in this lab. So like 4 things that are important that you do, or that you need to do in order for the lab to work out well. And again, I've watched this video, so I know what he says. So there's going to be another video. You have to write down 4 important sort of like parts of design of the lab that he's going to point out, and you're also going to see what the equipment looks like, so that you're ready to do the lab on Monday. All right, I will see you guys on Monday. Hope you enjoyed your Thanksgiving.